So today we're going to talk about the RH blood group system. There are two main blood groups that you need to know extensively as a immunohematologist, a medical technologist, a medical technician, a biomedical engineer, a clinical laboratory scientist. You need to know the ABO blood group, which is critical and crucial in transfusion reaction, and you also need to know the RH blood group system. So in this session, I just want us to do a brief overview as to the RH blood group system and how this complex system was derived. So we're going to go in brief details of the systematic design of this blood group system so we can have an appreciation as we go through our uh, investigation on the RH blood group system. So the RH blood group system, as we discussed previously, is highly complex. And the alloimmunization to the RH blood group antigen can be complicated in what we call transfusion reaction and also complicated in pregnancy. It is imperative for us to have a basic understanding of the RH blood group system, or let's say the RHD typing, which is a critical component in pre transfusion and prenatal testing. So, when a mother is of one RH group and the child is of another RH group, or the father is of a different RH group, that can complicate the whole dynamics of how the body of the mother reacts to the child. All right, but let us take it step by step as we get into the meat of the matter, the matter of the meat that you came here to enjoy. So, what I want you to understand and appreciate extensively is the term RH. Basically, it just refers to a specific red blood cell, RBC, antigen, we're going to call that the D, and to the complex blood group system, we have come to realize that there are approximately 61 different antigenic specificities that exist when we talk about the D. All right? So just bear that in mind. So, although the RH antibodies were among the first to be described by researchers, we in today's realm continue to investigate, continue to explore to try to have a better understanding of its intricacies and its complexities. Because this blood group system is so important that over time we are discovering more and more variants that exist with this blood group system. So research has been continuous and what we say ongoing. So from time to time, you may have new information being presented, and the knowledge will be shared as those information become available. So as we continue to explore these complexities of the RH blood group system, from its serology to its mode of inheritance, which when we talk about that, we're talking about the genetical control mechanism. We can also think about the biochemistry in terms of the structure of the RH antigen. So the RH is the second most important of the blood group system next to the ABO blood group system as it relates to the transfusion reaction. As the RH system, in this system, the antigens are very immunogenic. So they are very, what we call reactive in simple terminology. So, unlike the ABO antibodies that are routinely found in an individual who lacks the corresponding antigen, the RH antibodies are produced only after exposure to foreign red blood cells. All right, so usually it's based on an exposure that the body generates the antibodies, And when those antibodies are generated, then those antibodies can fight against whatever is inside of the individual and create complication. So we don't want to have a complicated system. We don't want our bodies to complicate matters. So we try to keep everything as simplistic as is possible. So understanding this will guide our minds to think about modern day, present day complications, present day challenges that exist. So when we're thinking about the RH, the first thing that I want you to think about is hemoblastosis fetalis, or I think that word is no longer being used, but in 2025 and beyond, I think the word that is being used is hemolytic disease of the fetus and newborns. That is HDFN, hemolytic disease of the fetus and newborn. Yes. So when we think about the RH blood group system and its implication in transfusion reaction, that is what I want you to think about. The term RH, capital R, common H, can be positive or negative. A person can be positive or negative, and it is just used to describe the presence or the absence of the D antigen. So if a person is considered to be Rh positive, it indicates that the person possesses the D antigen. So they have Rh antigen present, so the D is present. If a person is Rh negative, this individual does not have the D on the cell. So they lack the D on the red blood cell. So in understanding this, we can get an appreciation in simplistic terms that a person can be Rh positive, a person can be Rh negative. And that is the information that majority of the population would need to know. But because you are not majority of the population, you are a minority of the population, you have to go a little deeper into understanding the complexities and the intricacies associated with the Rh blood group system. But before we get into that, let us go down memory lane as we investigate some history of the Rh blood group system. So it was in 1939 that Levine and Stetson, who described a phenomenal, I think it was the hemolytic transfusion reaction, which was observed in an obstetric patient, right? A person that was pregnant, all right? But what they realized is that after delivering a stillborn infant, the woman required transfusion. Her husband, who was the same blood type as her, was selected. However, after transfusion, the recipient demonstrated classical symptoms of an acute hemolytic transfusion reaction, or AHTR. All right. So you must be saying to yourself now, if you are of this group and the husband is of this group and you got the same group, what kind of problem could exist? But that there is where it was revealed that the ABO is important, but there's another group that is also important, right? So subsequently, an antibody was isolated from the mother's serum that reacted both at the 37 degree, which would be the IAT phase, and the 20 degrees Celsius, which would be basically the room temperature phase, with the father's red blood cell. So it was postulated that the fetus and the father possessed a common factor that the mother lacked, which in some instances could be true for the cause of this situation. But while the mother carried the fetus, she was exposed to this factor and subsequently she produced antibodies that showed positive reactivity when tested against the transfused red blood cell from the father. So the antibody specificity was not at that time identified, but it was found to react with 80% of cells tested and was suspected to be a maternal antibody to a factor on the fetal cell. But as time would progress, their assumptions would have been tabled, and it would have come to a revelation that what they assumed had truth in it, but it had some things that needed to be ironed out. All right, but a year later, Langston and Wiener described an antibody made by guinea pigs and rabbits when they were transfused with rhesus macaque monkey red blood cell. So, rhesus monkeys. When we talk about the RH blood group system, we're talking about the rhesus blood group system. So, when we think about the rhesus blood group system, we're going to think about the monkey blood group system. It's the monkey that gave us these the antibodies that we could use to determine whether someone is D positive or they are D negative. So it's interesting how we came to discover and how we came to appreciate and how we came to use this information that we have. And it started from a simple situation where there was a mother who was given birth and she had a stillbirth, and then when the transfusion took place, then we realized that there was something else that could have been uh, uh, possible to cause that transfusion reaction, even though the blood groups were the same. So with that knowledge, we come to realize that something else could have been there. And we are very thankful for the work of Langston and Wiener, because they have been instrumental in us having the RH blood group system and the different classifications of them. All right. So as history would progress, we would come to realize later down a subsequent investigation by Levin and co-workers demonstrated that the agglutinates or the antibodies causing direct agglutination of the RBC causing the hemolytic transfusion reaction, and the antibodies described by Langston and Wiener appear to be different. All right. And maybe, just maybe, in that instances, they thought that maybe it was a different group. So, but many years later, it was recognized that the two antibodies were indeed different. However, the name RH was retained for the human-produced antibodies, and anti-rhesus formed by the animal was renamed anti-LW. And when we talk about anti-LW, it looks like the RH, but it's not really the RH. And this is the Lewis blood group that we're going to get into that. All right. So this anti-LW was in honor of those who first reported it, which would have been Lanston and Wiener. So the errors that came up gave us ideas, gave us concept, gave us an understanding of what was happening. It was giving us a guide. It wasn't clear, but over time, with more research, with more investigation, we came to realize, understand, and appreciate what was really taking place. And with that, we were in a better position in understanding exactly what was happening. So by the mid 1940s, five antigens were defined in the RH system. All right. In the 1980s or 40, years later, in the 1980s, we had molecular testing, which further defined the Rh gene, which gives us even more intricate information and details. So the Rh blood group system continued to be explored even today, even years later, because we're now in 2025 and moving forward into 2000 and beyond, and we're still discovering. So yes, there is still great work to be done. We're not going to know everything about everything. So as time progresses, we'll learn little by little and compound information that we have as we seek to maximize what it is that we know and become expert in the particular field. So let us now move into some terminologies which you may need to know, appreciate, and then we're going to look at the Fisher race terminology so that you can have an appreciation as we take it step by step in understanding the RH blood group system. So there are four terminologies used to describe the RH system. Two are based on the postulated genetics theory of the RH inheritance. The third common terminology used to describe only the presence or the absence of the given antigen, which is simple, easy. And the fourth was established by the International Society of Blood Transfusion, that's the ISBT, Committee on Terminologies for Red Cell Surface Antigen. So we have to come to understand that different groups classify them differently. So we are going to, in some instances, have to do a reverse engineering to figure out what is that classification in another group. All right. So it's important for us to distinguish between what we call the phenotype and the genotype before we explore the RH nomenclature. So when we talk about the phenotype of a given red blood cell is defined by the serological detection of the antigen using the specific anti-sera. So when we add the anti-D and we can detect the D being present, that is the phenotype, all right? So therefore the RH phenotype represents the result for the serological testing. And when we talk about the serological testing, we're talking about the D, the C, and the E. All right, so we have big D, small d, big C, small c, big E, small e. So that is basically what it is that we're looking at. And the genotype now. A genotype is an individual's actual genetic makeup. An RH genotype refers to the actual RH gene inherited from the parents, right? So your parents are important in your blood group determination. To depend on your blood group, your blood type, your parents would have been intricately involved. And we know that the father produces 50%, the mother produces, gives actually 50%. So 50 and 50 gives us 100%, which makes us a whole human being. So we have to decide what is in our best interest and if our parents are really necessary to our existence in this room. And I don't want you to think negatively about your parenting or your parents' contribution to who you are and what you have accomplished thus far, right? Your parents have been there for you. They have given you good things. They may have given you some genetic diseases, but guess what? Without your parents, you wouldn't be here. So let us just give some appreciation to the parents. Now, let's get back to the conversation. The first group that I first group in terminology that I want us to consider is the Fisher-Race terminology, which is the DCE terminology. It's simple, but I want you to get some concepts so that you don't confuse it with any other terminology. So this was discovered in the mid 1940s by Fisher and Race, which defined the 5 common Rh antigen and postulated that the antigens of the systems were produced by 3 closely linked genes. And the genes that were linked were D, C, and E. All right? So DCE. So each gene was responsible for producing one antigen on the red blood cell. So each antigen and corresponding gene were given the same letter designation, which referring to the gene, the letter is usually italized. So if you see the italized letter, that's a gene that is being referred to. So the Fisher and Race named the antigen system D, big D, or capital D, uppercase D, lowercase d, uppercase C, lowercase c, uppercase E, lowercase e. So now it is known that there is no lowercase d antigen, but some reference continue to use the lowercase d with the Fisher-Race terminology as a placeholder for D negative individuals. All right, so you understand that you have the Fisher-Race system D, C, E. Simple. So according to the Fisher-Race theory, an individual inherits a set of the Rh genes from each parent. So one set parents give you one set, the other parent give you another set, and then you combine, and then you get what is your actual nomenclature. All right, so the combination of the genes inherited from one parent is called the haplotype. For example, if one parent has DC, capital D, capital C, lowercase e, then the haplotype is written as DC, capital D, capital C, and lowercase e, and all combined. Simple as that. So the pairing of the maternal and paternal haplotype determines the offspring's genotype. So that is how your genotype is determined. So the Rh gene inherited from each parent can be observed when we do the genetics. All right, so the genotype is written as 2 haplotypes separated by a, what do you call that, like a slash, a dash? It's not a dash, it's a slash. Yes, it's a slash. I'm not remembering what you call the slash when you're typing on the keyboard, but that is what it is separated by. So you have a capital D, capital C, and lowercase e. That was the first parent. The second parent, capital D, lowercase c, lowercase e. All right, so from this now we can determine what the person's genotype is. But we need to remember that there are some exceptions to the rule, and there are rare phenotypes that involves deletion of specific genes, and in those cases the deletion is represented by a dash. All right, so that's simple enough for us to understand. For example, an individual having only D antigen and no C and no E, you would have D dash, and the dash serves as the placeholder, right? So placing the parent, when in some instances when you look at it, you may see it in parentheses. So the letters may be in parentheses. You may see D parentheses, C parentheses, and E in parentheses. Basically what it indicates is that this is a weak expression of the antigen being observed. All right, so what I want us to do is to just investigate a little bit about the fisheries theory. So let us get on the tablet and let us get the chalkboard ready for us to investigate. All right. So now what it is that we have is a system which has been made evident for us. And in this system, we have the phenotype, and then the phenotype gives us the designation. All right, so we are just going to have to work with it. So what we get as a phenotype, let's see if I can get this on the board. So we have the phenotype. Let me see if I can get the pen a little bit bigger. So the phenotype, reaction with the antiseria. All right, so what we have D, we have C, we have E, and we have lowercase c and lowercase e. All right, so when we have this, after we have this reaction, we're going to have the designation. The designations will be right here. So let us write it, designation. D-E-S-I-G-N-A-T-I-O-N. All right. So if we have D being positive, C being positive, E nothing, nothing, and that is positive. What we have is just write back exactly what we see, D, C, and E. So capital D, capital C, lowercase e. It's that simple. So don't worry yourself. If D is negative, just watch it. D is negative, we have C positive, that's big C, and we have negative big E, negative lowercase c, and we have positive lowercase e. Remember, D, there's no small d antigen, so we can just ignore the D, and we have the C, and we have the E. So we'll just put it back just like that, and that's just it. All right. So with this idea, we can go ahead and do another example. What if D is positive and C is not positive? Bigger E is positive, and let's say little c is also positive, and E is negative. So what we have here, D is there, we have E, and we have a little c. All right, but guess what? The representation is DCE. So because it is DCE, let's get back to it. Because it's DCE, we write the D, and then we look for the C, lowercase c, and the E is big E. So we do it like that, right? So we just try to keep the order the same as it is, DCE. All right, so next one, let us try another one. Let's say that we don't have any D, any big D, any big C. We have big E, we have little c, and we don't have small e. All right, so what we're going to have here is the D is not present, so we are not going to put lowercase d. We're going to leave that out, and we're going to put the E, and then we're going to put the C. So with this concept, we can understand exactly how it is that we can work out what is the designation based on the phenotype that we are receiving. I want you to realize what we have here. We have E and C, but it is written as D, C, E. So what we're going to have to do is just switch this up a bit. So it's going to be C, E. All right? Just remember that. It's DCE. Let's write it DCE. And this is for the Fisher race nomenclature. All right. Now let's try another one. So we have capital D, capital C, we have capital E, we have no lowercase c, no lowercase d. What we're going to get? We're going to get D, C, E, just like that. So as we go through these, you can realize that, hey, this is not so difficult. Let's try one more and see. All right. And then this is a designation right here. So this is not so difficult for us to understand and appreciate. What if we had no D and we had, let's say, big C, we had big E, we had no C there because we have big C and we have no E. So this here, in simple terms, would be C and E, just like that. And let us try two more and see if we can get our minds wrapped around it so that we can have a better appreciation. So I'm just going to write these two on the board in real time so that we can have them and we just run through them quickly. Because what if you got one of these? Would you be able to identify if you got one of these on the exam? So let us just try to get this, mean that you understand the concept of how we derived to this area. All right, so this is a phenotype here, Phenotype, and this is a Fisher race. So looking at this, let us see if we can find the designation. So this is big C missing, big E is missing, but let us look at what is there. Big E, big D is there, little c is there, and little e is there. So it's simple. Big D, little c, and little e. That's it. And then now let us look at the last one. We have big D absent, big C absent, big E absent, but we have little c present and little e present. So this here is going to be little c and little e. We don't have small d because the D antigen is not really a thing. All right. So now that we have that out of the way, we can express our knowledge and we have an idea as to how it is that we can derive a person's blood group system in terms of deriving the RH blood group system. So with that said, what are we going to do? We are going to use this information that we have garnered in order to create a mental framework so that when we go into the other nomenclatures, we can integrate based on what we already know about the RH blood group system. I just want to say thank you for sitting with us in this session, and we will be continuing on this journey because we're just at the starting block. There are a few more hurdles that we need to complete so as we can get the full understanding of the RH blood group system. Until we meet again, this is your professor signing out.